that the gametes are being formed, male gamete is formed, female gamete is formed, but there is one big difference between these gametes, and that is DNA methylation is also different in male and female gametes. Okay. So what happened during PGC migration, during the primordial germ cell migration? So remember that primordial germ cell, they are the part of the body, right? So, but they are set outside of the developing embryo. But they all came from either primordial, you talk about primordial germ cell, you talk about rest of the body, they all came by fusion of sperm and gamete, right? It means their chromosome should have some sort of methylation pattern. But when they migrate from the place of their origin, extraembryonic places, to the developing gonad, during that process, all the methylation patterns on DNA are removed. So that means when they are entering the gonad, they are kind of a clean slate. You guys know what is the purpose of methylation and all those things. You know about the epigenetic program and all those things, right? I think you guys also have studied those, right? So methylation patterns, they determine the activation or inactivation of the chromatin, how a gene is going to be expressed. So remember, like all the cells in the body, they have the same genome. There is no difference. But why some cells are like skin, some are kidney, some are cardiac cells? Because of the epigenetic patterns. And those epigenetic patterns are determined by the methylation patterns of the genome. Okay? So all those methylation patterns are removed when primordial germ cell they migrate to the gonad. It means there is no pattern. It means every gene is active in those cells, right? So that's why they have that power to become everything. But what happened when they enter gonad? So when they enter gonad, then what happened? That the gonad specific methylation pattern are written. When they enter the gonads, in gonads, gonad specific methylation pattern are established. So what does it mean? It means that the gamete, if it is sperm versus ovum, they will have very different methylation pattern. So that means what a sperm can do that ovum cannot do, because those genes are not expressed there. But what an ovum can do that a sperm cannot do, because of the methylation pattern differences. So what happened? There are essentially 3 steps. Number 1, the methylation patterns are removed. That's the first thing is happening. We are saying when primordial germ cell are migrating. Second is in testes. New methylation patterns are formed, and those methylation patterns are formed prior to meiosis. That means before puberty. But in female, what happened? DNA methylation occur after start of meiosis, and continues till puberty. So that means even the DNA methylation is also a very long process in the female body. So it's a prolonged process. Now, the set of processes, those are erasing and adding new methylation pattern. It means what is happening here, we are saying overall epigenetic reprogram. Epigenetic reprogram occurs during the formation of units. Okay? And it means that not only the structure, not only the cytoplasm, but the type of genes which are going to be activated in sperm and gametes, in sperm and ovum, will be very different, right? So, a lot of epigenetic reprogramming takes place. Now, this is the place, this is the step, this is the final step. They are produced, their genetic components are reprogrammed. Now, they are ready to fertilize. But this is the stage, think about, if you think philosophically, if sperm and egg, they are not going to unite, both are going to die. So these two cells are at the verge of death. But their reunion to rejuvenate them and start a new life. Right? So this was all about gametogenesis. Now we'll start our next topic. So we are going to talk about so remember that the developmental processes are very complex. So we will start with a simple example, then we'll move to mammals, right? So we are choosing the sea urchin that is well studied fertilization, because external fertilization. Then we'll talk about what happens in the humans, okay? So let's start with external fertilization in sea urchin. So if you think about sea urchin, or sea urchin live in the ocean, right? And all the animals which are basically doing fertilization in the water, they are laying their eggs in the water, right? But think about, there is not only sea urchin, other fishes are there. Sea urchin will also have thousands of species there, right? So it's a, what is happening when they are doing? So we are saying that the gametes, they are released in pool or ocean. They are released there. But then what is going to happen? So other species also do the same thing. If sea urchin is doing, the other species are also doing it, right? So then there are two problems right there. One problem is that immediately when you put those sperm in there, what happens? They are diluted. They are diluted. because ocean is so big, pool is so big, sea urchin is like, they're all diluted right there, right? And then you can think about that if they are diluted, then how sperm and egg can meet? How they can meet if they're so diluted? And then if there are so other species also there, then how can sperm and eggs are prevented from being fertilized by other species? How that happen? Now these problems, these are big problems, right? But these problems are circumvented by two mechanisms which exist in these species. And those mechanisms are, number one, there is species specific sperm attraction. And then there is species specific sperm activation. So that means egg has that power. So the egg can attract only the species specific sperms, right? And the other thing is that the egg not only attract the sperm, but also activate the sperm so that it can do the fertilization. Okay? So then we are saying how that happens. So these sperms, the sperms are attracted to the eggs by chemotaxis. By chemotaxis. That means eggs secrete a chemical. Chemo means chemical, taxis means attraction. So attraction by a chemical. Okay? So that means sperm, they follow the gradients of the chemical secreted by egg. Okay, so the eggs, they secrete certain chemical, and the sperm can sense that chemical and sense the gradient, going from lower concentration to higher concentration. That is making sense so far? Now, the other important thing is that eggs did not only attract the sperm, they actually also control the timing of the fertilization. So egg attract the sperm and control the timing of fertilization. So if you take, for example, this is one experiment, this is a slide, for example, okay? So if you take here, put the sperm here, put the egg here. Now this egg, for example, is in first meiotic phase, okay? It had just completed the first meiosis and started the next cycle. Or it is even before that, just primary oocyte is there. But here you have an egg that had just finished second meiotic cycle. So that means that has just, and that has become haploid, okay? In sea urchin, fertilization takes place after the second meiotic age, right? In human, it is still in second meiotic metaphase, we know that. Here, if the sperm is put here, when they have just finished this thing, only then this sperm can go toward this. Before that, this sperm cannot go toward this. It means that oocyte or egg, it releases those chemicals only when it is ready for fertilization. It doesn't mean that if the sperm is there, it will just near the egg, it will just go and fertilize it. No, it will not do. It means that ovum is going to determine the time. It's going to release certain chemicals. Those chemicals are going to tell that, hey, now you have to basically start the fertilization process. That is okay so far? Okay, now. So now we want to talk about that how the sperm acquire the motility. So, motility and direction of sperm. So, sperm should start moving, that's the first thing. And then sperm should know where to go, right? Because it's an ocean. So, the first thing we are saying that all those things are controlled by chemotaxis. And chemotaxis means certain chemo, it means certain chemo attractant molecule should be secreted, right? But chemoattractant molecules are very different even in the closest species. So the chemoattractant molecule, they are essentially determining which species. So chemoattractant molecules secreted by species A will be very different from the species B. Okay? So that's important thing. Now what happened? The sperms in the testes are not capable of any movement. They're not capable of any movement. The reason is there is low pH, around 7.2, and this pH is low because of high carbon dioxide. So in the blood, there's high carbon dioxide during that area. So that means you make more bicarbonic acid, you have more acidity, right? And when that happens, what that does, so now the sperm are released in ocean. Okay. So ocean is, there is seawater. Seawater is more basic, right? So seawater raises the pH. And that pH goes up to 7.6. And when the pH go 7.6, what that does, that activate dynein ATPase. That activates dynein ATPase. Now, if the dynein ATPase is active, that means you are splitting ATP. Therefore, splitting ATP. Therefore, energy to move and movement. And remember, where is dynein ATPase? Where is dynein ATPase in the sperm? Where? Axoneme. Where is axoneme? Flagella. That's what I wanted to hear. In the flagella. So flagella movement start because flagella allowed to move, right? So the movement is now acquired, but direction, where to go, right? So movement is there. Movement is there, but direction is not there. So movement doesn't provide direction. So where to go? So if you are saying direction, that means this sperm should be able to now find out that chemoattractant, right? So that it can go toward that. So then there are, so going from egg, just to remind you, sea urchin egg. Okay, so this is called white line animal here, right? And all the fishes, they also secrete a egg jelly. So this is the egg jelly. So they are secreting some egg jelly. Okay. Now, these egg jellies, they secrete. So egg jelly secrete a small sperm activating peptide. Sperm activating peptide. Okay. And this sperm activated peptide in the species called Arabica punctulata. Just one species, just in one of the species. You don't need to remember the name of the species, right? In one of the species, a small, the name of this is called resect. R-E-S-A-C-T, resect. So resect is a sperm activating peptide. It's a small peptide, so this is just made up of 14 amino acids. 14 amino acids is there, right? So now resect from this species only attract the sperm from this species. It's not going to attract sperm from any other species. So that's a species specific difference. So they are very specific. So the resect or the sperm activating peptide named resect, which is secreted by Arabica punctulata, will only attract the sperm from Arabica punctulata, no other species. So there is species specific differences. Okay? Then what happened? How it works? So we are saying on the sperm membrane, so this is, I'm drawing the sperm membrane. So this is your sperm membrane, right? On the sperm membrane, and when I'm saying sperm membrane, I'm saying the area where the flagella is. Okay? So this is the flagella or tail sperm membrane. This is in the tail area. This is in the tail area. So in the tail area, there is a receptor. So this is the receptor there. Receptor for resect. This is a receptor for resect. Now this receptor is also an enzyme coupled receptor. If you remember from cell signaling, we said there are some receptors which have the enzymatic activity. Okay? So this is So this is an enzyme coupled receptor, and this is called, I'll write here the short name, RGC, receptor guanylyl cyclase. Receptor guanylyl cyclase. Okay. Now this receptor guanylyl cyclase, what it does, so now this receptor is there, and say this resect come, so this is your resect, it come and bind here. Okay. Binding of this activate the enzyme coupled receptor. And when that happens, what it does, when that is going to happen, it's going to convert a GTP molecule into cyclic GMP. So GTP is converted to cyclic GMP. And what happened right there next to this, this is a calcium channel. Right there is a calcium channel. Now what happened, when this cyclic GMP is made, there is a receptor right here, or a binding site for cyclic GMP. So this cyclic GMP is made right here. This cyclic GMP go and bind to its binding site, the cyclic GMP is there, go and bind to the calcium channel. And binding of this open the calcium channel. So if I'm writing the step, I'm saying number one, resect binds to receptor guanylyl cyclase. Okay. Second step is GTP converts to cyclic GMP. Third step, cyclic GMP binds to calcium channel. Okay. And when it binds to calcium channel, what happens? This calcium channel opens. So the fourth is opening of calcium channel. So when the calcium channel opens, what happens? That leads to calcium entry in the cell. Okay. That's step number 5. Calcium entry the cell. Now this huge amount of calcium and increase in GTP, it does two things. Number one. So two outcomes of this whole thing. Increase cyclic GMP and increase calcium. What they do, they activate ADP production machinery in the cell. Here it is middle piece, right? And the second thing they do, increase in calcium, activate dynein ADPase. And when the cell is saying, what is the whole purpose of life? Energy, right? That's what we eat food, to get energy, to work, to all those things, right? So when it is getting that boost of energy and energy making mechanism, what it does, it essentially swims to. So it follows that gradient. It says, okay, from where there is more, we need more ATP. So more ATP is coming from the egg, because egg is making reset, so it follows that gradient and move toward the egg. That is making sense so far? So we are saying, essentially, during this fertilization, the different species, they can secrete, they can not secrete, they can spawn their gametes in the ocean, but they can be diluted, and that dilution problem and distance problem is controlled by the chemicals secreted by the egg. And they act as chemoattractant, so there's chemotaxis phenomenon taking place. And we are saying that egg itself controls not only what is going to attract, but also the timing of the fertilization. And then we are saying the sperm, when it is released, it doesn't know how to move. It also doesn't know where to go. It doesn't know how to move because in testes it cannot move because of high carbon dioxide concentration. It has low pH. And low pH enzymes don't work, right? But when it comes to the ocean alkalinity makes the pH 7.6, 1980 pH is active, and 1980 pH is active, that means the flagellar movement can take place. So sperm attains the power to move, but does not have the cue for direction. That direction cue comes from sperm activating peptides, and those peptides are secreted by the egg jelly. So egg jelly is secreted, and those peptides are very specific to the species they are meant for. Here we are giving example of resect. Resect is made in Arabica punctuata, 14 amino acid peptide, very small peptide. That binds to its receptor on the flagellar membrane, or the tail part of the sperm. When it binds, it binds to receptor. Receptor is a enzyme coupled receptor called receptor guanylyl cyclase. When it binds there, this receptor is activated. That means the enzyme associated with it is activated, and that converts GDP to cyclic GMP. So you have more and more cyclic GMP is made. That cyclic GMP come here. So this is the step number 1. This is the step number 2. Cyclic GMP bind to cyclic GMP binding site on the calcium channel. And that binding open the calcium channel, and calcium start getting in. And when calcium get in, both the increase in calcium and cyclic GMP, they activate ATP production machinery in the mitochondria. That means they supply the essential energy. So now they also further activate the calcium, further activate 19-ATPase. So you have more powerful thirst of the flagella, and the sperm knows from where that signal is coming, and then sperm start moving toward the egg. So sperm basically move, flagella move like this, and basically they assess where that is. So flagellar movement kind of scan the area around and find out where to go. That is making sense? So talk to each other for a minute, then we'll go further. to secrete it.
fertilized by others.
That's how they're going to control it.
So there's
If they secrete it early, then
something
they
to
will
do,
slow.
and it's built by the egg.
So it's
So
only
there's
secreted when the egg
species-specific
is
attraction and for sperm
Yeah,
attraction.
and jelly is
And
because jelly is
species-specific
also
sperm
secreted by the egg.
activation.
But whatever is surrounding the egg is coming from, like white light was
And
egg is
attracts
jelly and
people
is
time to fertilization.
Right.
So the egg can fertilize after the second gatets. So then sperm are attracted to egg and fertilize, and sperms follow the gradients and become the same pattern. Motility attraction is the chemoattraction non-holistic between species, and the sperm and the passenger not control movement because of the low pH of the CO2 because of the high CO2 levels. Sperm are released in the ocean, the pH is raised to 7.6, the diamine 82 days, so where do they go? And then the egg has jelly, big jelly that's small sperm activating peptide. So then we look at the species for wherever and they do RSAC, which is a 14 amino acid peptide. It's only for that species, it's not the species except on the sperm tail membrane, there is a RGC receptor that the RSAC binds to, which causes the conversion of GTP to cyclic GMP, and then cyclic GMP binds to the calcium channel, which causes the calcium channels to open, which causes the calcium to enter the cell, which increases cyclic GMP and or calcium activation, ATP production machinery in the cell, and that's in the mitochondria. And then it also increases the calcium activated diamine species, which causes the sperm to split.
Any question? Any question here? Question? No? So, now what has happened? We are saying that this is your egg and word line envelope and your egg jelly is there. And it has attracted the sperm toward it. Now sperm has actually reached you. So think about this, the sperm. That sperm has reached you to the egg jelly, right? Now what should happen? Now sperm should be able to essentially, so if this is the egg jelly there, so this is the egg jelly there, sperm should be able to move into egg jelly, then it should be able to cross white line envelope, then it should be able to fuse the membrane of the egg. Right? Sperm membrane should be able to fuse the membrane of the egg so that it can pass the genetic material. Only nucleus is going to go, right, inside this egg. So how that process is going to happen, right? So the first thing that happens is that when the sperm is reaching the egg, it just touches the egg jelly. So interaction of sperm with egg jelly, what that does, that causes a process, and that process is called acrosome reaction. Jelly cause acrosome reaction. So what is acrosome reaction? Acrosome reaction has two components, and I'll explain those. So remember, this was our sperm. This was the nucleus right here. And then we said, right here, was acrosome vesicle. Right, let me draw with different colors so it will be more easy to understand. And this is your sperm membrane. Right? And right here, we said there was G actin. Globular actin was there. Okay. So what happened? When this sperm comes in contact with the egg jelly, two steps happen. Number one happen is the fusion of acrosome vesicle with sperm membrane, and that leads to exocytosis of acrosome vesicle. So, what is going to happen? So this acrosome vesicle, if it's a vesicle, exocytosis means should move forward, right? Then it means that this should kind of fuse with this, that lead to something like this. This is a GFN. That's how exocytosis occurs, right? Vesicle go and fuse. And then you have all this component which were in the acrosome vesicle, they fuse. So what is this? These are proteolytic enzymes. Proteasomes are also there. And other digestive enzymes. So they are all present here. They will be all released out. Making sense? Now, if they are being released out, that means they can digest the egg jelly, right? So proteolytic enzyme, they all are going to digest the egg jelly. That is going to allow sperm to move forward. But then what happened? When this is released, something else happened. And that is what happened, that this G-actin, remember when we're talking this in the sperm structure, we said this G-actin is going to make a microfiber, right? Change into fiber actin. And what happened, the G-actin changed into the fibrous actin, and what it does is, essentially, it starts making like this, a fiber, like this. And that kind of push it up. So F-actin changes to G-actin, and you get, because of that, you get actin, microfibril.
It's the other way, right? G-actin makes that.
Oh, it's the other way. G actin change to F actin. Global actin change to fibril actin and make it actin microfibril. And what happened? That kind of push it up. And what is going to happen? That is going to form a structure like this. Right here is your F actin. And right here is your sperm like this. Understand this? So, something like this is going to form. Now, this is called acrosome process. Acrosome process. Right? And this acrosome process, this acrosome will be actually, on this will be attached right here. You will have the proteasomes attached to it. And what these proteasomes are going to do? That is the first thing happen. Fusion of acrosome vesicle does this. Second thing is G actin change to F actin, right? And that cause formation of acrosome process. Okay. So these content of acrosome vesicle, they are going to digestive path through the matrix. Now, think about, this is the next step, this thing. So this acrosome process is now formed, and this acrosome process has proteasome here attached to this, right? And this acrosome process is now attached to white light membrane, because that's what happened. So you have basically digested the matrix by the content of acrosome vesicle. Now acrosome process is now attached here. And what will do? Acrosome process is going to digest through, because it has the proteasomes, to the white line envelope and basically attach the sperm to the egg membrane. So you're going to the egg membrane. Making sense? So acrosome process with its attached proteasome is going to digest through the white line envelope, and essentially it is proceeding toward the egg membrane. Making sense? So this process, this whole thing is called acrosome reaction. So you guys are okay with this? So talk to each other for a minute, then we'll go further. Okay, any confusion here? You guys are okay? We're fine here? Okay, good. Now, what is happening there? We are saying that, this is an interesting thing, just think about it, that you have an egg here, this jelly is there, it has invited a sperm here. Now we are talking about acrosome reaction in depth, okay? So what caused that acrosome reaction? We said that sperm is attracted by reset, right? But then it is there, but then what caused the acrosome reaction? And we are saying that again, that is done by egg jelly. So egg jelly has sulfated polysaccharides. The sugar which have sulfur in them, right? So they have sulfate containing polysaccharide. They secrete those. So egg jelly has sulfate containing polysaccharide, they are secreted. And they are secreted right here when the sperm is in the vicinity. Now think about, this is one species egg. Its sperm was somewhere there, it invited there. Maybe there is another sea urchin right there close by that has secreted its sperm right there, nearby. So don't have to attract them, right? But they are in the ocean, so they have the movement, but they are right here. So you will say that these sperm should also undergo acrosome reaction, right? But that never happens, because these sulfated polysaccharides are also very specific. So if a species secretes this sulfated polysaccharide, that is only for the sperm of that species, no other. So that is another barrier. What we say that this is kind of a species specific barrier against cross fertilization. Because they are very specific. Now, if we look here, so this is an example. So they are taking those sulfate containing polysaccharide from different species. So if the sulfated polysaccharide from A. luxula species, and you have A. luxula right there, you will find there acrosome reaction take place only in the A. luxula, nowhere else, right? But if you take here this sulfated containing from A. leucanter, only A. leucanter shows acrosomal reaction, no other species. Same with A. variegatus, L. variegatus. Again, only L. variegatus show, no other. So that means that even the closest species, even the sperm reaches the egg, it cannot initiate the process of acrosome reaction. And if it cannot initiate the acrosome reaction, then acrosome vesicle will not exocytose. That means even if the sperm is here, it cannot digest the egg jelly, and then there is no acrosome process, so it cannot digest the white line envelope. So that's why these are barrier. These secret chemicals, they also act as barrier against the cross species fertilization. That is making sense? Okay. So we will talk about mechanism, but I cannot finish mechanism in the next 3 minutes, so that will take more time than that. So we'll stop it here. And so good news is the department